All right, guys, today we are going over some notes with DNA replication, and then I'm going to walk you through what you are doing for your lab this second half of class. If you do not finish the questions on the lab, that is totally fine. Just make sure that you are keeping that lab, and then we will finish it when I get back tomorrow. All right, so looking at cells and how DNA actually replicates. Before a cell divides, we have to replicate that DNA. So it's going to happen during that S phase of interphase, as we have talked about many, many times. The structure of DNA itself lends itself to replication. So if you remember, these backbones on the DNA are very, very strong. Okay, so they are repeating units of that phosphate and that sugar on the backbone. And then the base, those are in the middle. The bases are connected to each other with relatively weak hydrogen bonds. Hydrogen bonds are really, really sticky, but they're easy to kind of stick and unstick. So I like to think of them like Velcro or like a zipper or something like that. The first step of DNA replicating itself is initiation. There's only 3 steps to this process. So this first step is initiation. There is an enzyme named helicase. So we talked about the other day that when we have a word that ends in OSE, ose, that is a sugar like lactose, glucose, fructose, anything like that. If we have a word that ends in ace, that indicates it's an enzyme. So if we remember from things we've talked about previously this school year, enzymes are made of proteins and they're basically like little cellular workers. So they are doing all of the jobs for us on those cellular levels. So our first step, initiation, there's an enzyme named helicase. You see the word helix, helicase. It unzips the double helix. So this is kind of what it looks like. So what it does is on our original DNA strand, it will break those hydrogen bonds and it creates area that we can actually attach new bases to. Our next step of this process is elongation. So we have another enzyme that works for us called polymerase. So poly means many, mer means molecule. So it's a many molecule, ACE is an enzyme, a builder, so a many molecule builder. What DNA polymerase does is it adds complementary nucleotides. So we remember that A always matches with T, G always matches with C. That is the one-track mind that this enzyme has. So as we can see, this little enzyme is chugging along and adding in complementary bases. Same thing on this side. And they're traveling in opposite directions because if we remember, DNA is the sides are parallel, but they are anti-parallel. So they are going in opposite directions, just like cars on a road. So we can think of these DNA polymerases like cars on each side of a road, where this one is traveling in this direction, adding new bases, and this one is traveling in this direction, adding new bases. The last step of this process is termination. So when DNA is replicating itself, sometimes we have little gaps where this new backbone that got added in might have a little chunk missing or something that's not quite connected. So what we have is another enzyme called DNA ligase. Okay. This word, a ligand, means to connect. So DNA ligase is basically like a little welder. And what it does is any little gaps that existed in that backbone, it goes zzz, 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 and it welds them together to make sure that our DNA is nice and strong. So it seals those newly synthesized strands, and then that completes the synthesis process of our DNA. This model of DNA is also called semi-conservative. Once again, breakdown of a big word. Semi means partially, and conservative means to save. So partially saving. Semi-conservative means partially saving. So if we flip back and look at the picture that we had of our DNA replicating with that DNA polymerase, we can see that half of this strand, so the light gray backbone, that is our parent strand, that's our old DNA, and it's adding this dark gray, that is our new strand, okay? So it's half old, half new. Every time DNA replicates, it unzips, and the new strand is half old, half new. So we call that the semi-conservative model of replication. So what you guys are going to be doing today in your lab is going through the structure of this DNA. So on your lab, in the little baggies that you have on your tables, let me just go grab one, hang on. All right, so on your lab tables, you guys will have 2 strands of little pop beads. So the picture up above is what they look like. So if you notice, this one is kind of facing that direction, and then there's an extra little red one over here facing the other direction. Those are the phosphates, okay? And then we have all the bases in the middle, and those are color-coded according to this. So what you guys are going to do to start off with is take out the 2 full strands that are in your bag. Please, for the love of all that is holy and unholy in this world, do not take these strands apart, because I have another class that's doing this and I'm not here to put them back together. Okay, so do not unpop these, please, 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 please. Okay, so if you see, you're going to line them up just like it is in the picture up above there. And then on this picture on the front of your lab, you are going to color code it so it looks just like the picture up above. And then you are going to label those different parts on your picture. The next step of what you're going to do after you answer the questions on the next page is you are going to go through and model the process of replication. Okay, so you are going to model the process of replication. So I'm going to show you in the next slide what this is kind of going to look like on your paper or on your table with the pop beads. And then you are also going to draw it in your lab and answer the questions that are in your lab. So what this is kind of going to look like, your first step of replication is you would have initiation. So DNA helicase would come and unzip your DNA strand until we have 2 lone DNA strands. Then you would model DNA polymerase coming and adding new bases by adding in new complementary bases. So we're noticing that green always matches with yellow and orange always matches with blue. When you are adding your new bases in your bag, you have little lone, oops, oh my gosh, where's my camera? Little lone nucleotides like this. If you notice in the picture up above here, you do not need to snap them together. Please don't snap them together so that way it's easy to clean up and set up for the next class, just because I am not here today, okay? As you're going through that process of replication, so we're adding in new bases on this side. On the other side, you are adding in new bases as well until you should get 2 strands that look the same as each other. Okay, so these 2 strands are going to look the same as each other, and that is what you are going to draw in your lab. So it says like draw the process of replication. So figure out how you are going to draw that process and then answer the questions at the end of the lab. So hopefully this helps with what you are doing to just kind of go over that process of DNA replication and that structure of DNA one more time.